Welcome back, everyone. I hope everyone had a chance to stretch and maybe get something to drink or eat, and excited to proceed with the afternoon, the rest of the afternoon. It's my absolute pleasure to welcome Elizabeth Head from the University of California, Irvine, who's going to talk to us today about cerebrovascular pathology as a function of age in Alzheimer's disease and Down syndrome.
Great, thank you. I'll just get my screen shared. So I wanted to start by just saying thank you so much to the organizers for the invitation. I'm pretty excited to talk about some of this work, and it's been really fun to see everything discussed on a molecular level, and I'm going to take it all the way up to human brain and a little bit of information about clinical features of Alzheimer's disease in people with Down syndrome. So just to start us, I'm going to talk a little bit about Alzheimer's disease in people with Down syndrome, just to kind of get us all on the same page. And then I'm going to talk through why we're so interested in cerebrovascular pathology. And then I think relevant to the topic of this whole meeting is why is it important to think about cerebrovascular pathology and how it might impact glial biology as well as neuronal biology. People with Down syndrome are living much longer than they used to be. There's been an almost fourfold increase in average life expectancy since 1970. And during those times, people with Down syndrome didn't live past really 15 years of age. And so this is terrifically good news, and that's partly due to improved medical care and managing co-occurring illnesses. But I think it's also related to access to better enrichment in terms of lifestyles and education. Interestingly enough, the most rapidly growing segment of people with Down syndrome are those people between the ages of 40 and 49 years. So, and this age epoch is going to become more important as we go through this talk, because this is really where we see the highest risk for Alzheimer's disease neuropathology being built up in the brain. So this means also in future, we need to start thinking about how to take care of folks as they get into these older ages and may develop dementia. However, I would also note that this life expectancy increase is not applied to all ethnicities and minorities. We still also have a diversity problem for people with Down syndrome. As most people know, Down syndrome does have three causes, but the majority of people with Down syndrome have a full extra copy of chromosome 21. And as a consequence, there are a number of proteins encoded on chromosome 21 that are overexpressed. And for example, one that is, of course, a big focus for a lot of us working in the area is that the amyloid precursor protein is on chromosome 21. And that leads to lifelong overexpression of not only APP, but cleavage into beta amyloid, which is one of the toxic proteins involved with Alzheimer's disease. But I would also point out there's lots of other genes on chromosome 21, so the story is never ever simple. In fact, I think that enriches our ability to understand Alzheimer's disease by focusing in on chromosome 21 for people with Down syndrome. Because of that extra A beta production throughout the lifespan, there is an age-dependent deposition of beta amyloid in the form of plaques in people with Down syndrome. And we typically see diffuse plaques really become systematically observed in people who are in their 30s with Down syndrome. These get progressively worse with age until around 40 years of age, where we really start to see the tangles start to kick in. So there's about a 10-year period where we see A beta deposited and then tangles kick in. And then I'll talk about what that means for cognition in just a minute. But another feature of this excess beta amyloid production is the deposition in association with blood vessels. And I put some images here from A beta 1 to 42 immunostaining in the frontal cortex of one of our brain donors with Down syndrome, and one of A beta 1 to 40, which is the more soluble form, which loves cerebral amyloid angiopathy. And you can just see how caked on that beta amyloid protein is on these blood vessels. So what this means for people with Down syndrome is by the time a person is 40, if they are full trisomy 21, they will have sufficient neuropathology in the brain for a neuropathologic diagnosis of Alzheimer's disease. But what does this mean for cognition? And what is the age of onset of dementia in people with Down syndrome? There's lots of beautiful data out there, different cohort studies around the world, small studies, big studies. The studies are getting bigger, longitudinal, and much more definitive. But what is strikingly consistent is that a lot of these cohort studies, including our own study in the Alzheimer's Biomarker Consortium, which I'll tell you a little bit about later, between 50 and 56 years of age tends to be the median or average age of onset of dementia in people with Down syndrome. And notice that this is at least 10 years after when we see full-blown AD pathology. But importantly, we are seeing a subset of individuals that get into their early 70s without signs of cognitive decline, and that isn't related to being mosaic or partial trisomy. And there is a range, an individual range of age of onset. So there's a lot of work in the field now trying to understand what are the factors that are driving these individual differences. Are they exposures, lifestyle exposures? Are they genetic risk factors? So there's lots of active and very exciting work going on in this area. So why are we so interested in cerebrovascular pathology in people with Down syndrome? The story is really kind of interesting. But again, I'll bring you back to this picture of what beta amyloid looks like on blood vessels and how it's essentially caked on these blood vessels. And you can envision with that much pathology that those blood vessels can't constrict or dilate properly. And there's going to be some breakage and leakage and loss of blood flow in certain parts of the brain. But there's this interesting tension when we think about vascular pathology in people with Down syndrome. They have protective factors, systemic vascular protective factors. They typically do not develop atheromas or atherosclerosis. And there's very little hypertension in these individuals. In fact, they tend to be more hypotensive. And that doesn't change much with age. So you would think these things are really good for their brains. But in contrast, although this is much, much better now, older individuals with Down syndrome certainly, generationally speaking, tend to be more obese and overweight. Part of that might stem from a higher prevalence of hypothyroidism. There is a very, very high prevalence of sleep apnea, which doesn't always get diagnosed and is apparent even in infancy. And that's because of the structure of the neck and the tongue. But also we have this lots and lots of cerebral amyloid angiopathy. So that kind of drove a question for us. Well, we've got two things going on here. We have a set of protective factors, but we also have a number of factors that potentially could really drive cerebrovascular pathology. So, and are these events occurring in Down syndrome to a greater extent or a lesser extent than what we see in late onset Alzheimer's disease in the neurotypical population? The way we answered this question was through a collaboration with Michael Phelan, who's a biostatistician here at UCI. And we queried every single autopsy case we had collected through our Alzheimer's Center here at UCI, which included 32 cases of people with Down syndrome, 80 with late onset Alzheimer's disease, and 37 age-matched controls, age-matched to the late onset Alzheimer's disease cases. And we dove into the neuropathology data we typically generate for every person here through our Alzheimer's Center that gets uploaded to the National Repository. And we focused in on vascular outcome measures to look at these hypotheses. And what we found was really pretty interesting. So when you look at this graph, we have Down syndrome, Alzheimer's disease, and controls, and the percent of people affected by this form of pathology, and whether there was mild, moderate, or severe. So darker colors is more severe pathology. When we look at atherosclerosis, we see that people with Down syndrome, as we had predicted, had less frequent and less severe atherosclerosis, and this is in the circle of bones. Interestingly enough, arteriolosclerosis was absent in this cohort of 32 people and pretty comparable for our late onset Alzheimer's disease cases and our controls. But again, as we hypothesized, the extent of cerebral amyloid angiopathy was far more frequent. It was far more frequent, far more severe in people with Down syndrome when we compare that to people who have late onset Alzheimer's disease. So yes, indeed, we have less atherosclerosis in the brain, but we have significantly more and severe cerebral amyloid angiopathy in Down syndrome. And then when Dr. Phelan kind of broke this out by ages, and this spread looks a little odd with the ages, but it distributes our cohort ages, we can see that with increasing age, the absence of CAA decreases, and the severity, the presence of more severe CAA increases in a linear fashion to some extent with age in people with Down syndrome. And it tends to be over 50 years of age. So we think about plaques in their 30s, tangles in their 40s. We have vascular changes really kicking in their 50s, which goes along much more tightly with some of the clinical changes we might see. So that brought the next question of what are the consequences of having this increased frequency of cerebral amyloid angiopathy? And we thought this most likely might be leading to more frequent microhemorrhages in the brain. And the way that we tested this hypothesis, again, was using human autopsy cases. And Alex Hellman, who was a graduate student with me at the time, very carefully quantitated the extent of microbleeds by using Prussian blue staining in tissue sections. And every time she identified a blood vessel, she would then count that as a bleed. And she did this for a lot of cases. And here's what she found. And we did this in frontal cortex in 50 cases in total. So when we look at people who are neurotypical, who are young, under the age of 40 years of age, she very rarely, if any, on average, did not find any microbleeds. If she looked at middle-aged neurotypical controls between 40 and 60 years of age, she found a scattering of microbleeds. And these microbleeds could have occurred at any point in the lifetime or prior to death. Old controls did not show significant increase in the number of microbleeds, and these were people over 75 years of age. Not surprisingly, sporadic Alzheimer's disease tissues show a significant number of microbleeds. People who with Down syndrome who are young, under 40 years without AD pathology, very infrequent bleeds. And those who had died with Alzheimer's disease and Down syndrome, we had also a very significant number of microhemorrhages in their brains. Although it's trending towards more microbleeds than what we see in Alzheimer's disease and late onset Alzheimer's, these differences weren't significant. So that was really interesting to us, and it's another pathway we're trying to kind of map down. We have so much more CAA, yet we don't have significantly more bleeds. And so that might suggest there's some factors on chromosome 21 that might be protective with respect to some of this vascular pathology. So I'm pretty excited about what we're going to be doing next. One is, and you'll hear from Adam Brickman tomorrow, is Adam and I have gotten an award to look at cerebrovascular pathology in a much more systematic way in people with Down syndrome, which would include an imaging component, looking at autopsy samples, and also collaborating with Donna Wilcock to look at biomarkers, fluid biomarkers. So we are just starting this project now. But as a little teaser, and some of the preliminary data we put into this grant, is how we're going to map down what's causing these microhemorrhages and what other aspects to cerebrovascular pathology are in play in the brains of people with Down syndrome, so that we can think about later on what interventions should we be thinking about to stop all of this pathology from happening. And the lead person in my lab who's working on this project is Dr. Lisi Flores-Aquilar. She joined me in January as a postdoc, and she's working with an undergraduate student, Kelly Poon. And what Lisi has done is developed a set of hypotheses based on this schematic, where she's going to examine the different cell types that are involved in the cerebrovascular unit and protein markers that might reflect damage to any of these cell types. So to that end, one of the first projects she looked at was just to look at the basement membrane in autopsy cases of people with Down syndrome who died with Alzheimer's disease. And a good marker for that is collagen 4. And what you can see, which I think is really striking, is when you look at controlled brains, we have lovely basement membranes on these blood vessels. They look very intact. In contrast, when you look at those with Down syndrome and Alzheimer's disease, and trust me, I enhanced the contrast, there's very little signal here. And she confirmed this with light microscopy. Again, here's a control case, beautiful blood vessels she sees stained with collagen 4. But when you look at the brain of a person with Down syndrome, you can see a couple of interesting features. One is the intensity is far lighter. But second, when you look at some of the morphology of these blood vessels, they look tortuous. So there's a number of parameters that Lisi is going to measure and quantitate from these samples to help us understand how this is contributing to vascular pathology. Another cell type that she's looking at are parasites, and parasite signatures are coming up a lot in single-cell RNA-seq studies of human brain, that parasites in these as vascular cells are compromised. And she's got some beautiful labeling now for parasites, and an arrow points to these parasite cells sitting on the blood vessels, extending and wrapping around that blood vessel. And when she does some immunostaining with this antibody against PDGFR beta, which recognizes parasites, she can see controls show beautiful labeling, so lots of intact parasites. But in Down syndrome, this is far more scarce, and it's certainly not as beautiful as what we're seeing in the control cases. So this is giving her a hint that parasites are also impacted with Alzheimer's disease in people with Down syndrome. And so I think the story is really going to be enriched by some collaborations we have with Jackie Yang and Manolis Kallas there at the MIT, and also Vivek Swarup here at UCI. There's a number of really beautiful single-cell RNA-seq studies coming out. So I'm hoping we'll have a much better understanding of all of the pathways that might be engaged in this process from the RNA level that we can take back to the protein level and take a look in human brain. So I think I will just kind of wrap this back around to why I think this might affect, these vascular changes might be important for glia biology. So as I talked about, there's extensive CAA in people with Down syndrome. There's lots and lots of microbleeds. We think there's some evidence of parasite loss, or certainly the proteins associated with those parasites. And there appears to be basement membrane disruption. So when you put that all together, it suggests to us that there must be serum proteins leaking into the brain that might be causing lots and lots of neuroinflammation. So that's probably something that's happening as we see a lot of microbleeds with increasing age. But we'd also note that there are at least a dozen genes on chromosome 21 that are directly related to inflammation. So there's probably also another lifelong neuroinflammatory phenotype, which we actually have shown in work with Donna Wilcock, that shows that people with Down syndrome are different than neurotypical young people in terms of their neuroinflammation. And another little note I'll put out there is chromosome 21 contains the S100 beta gene, which is a protein that's expressed in astrocytes. So overexpression of that protein might also impact how astrocytes function in the Down syndrome brain. So if we hypothesize those serum proteins are getting in and are driving inflammation in the brain, we of course would want to look at the usual suspects, which are microglia and astrocytes. And there's some really beautiful work characterizing microglial phenotypes and their molecular pathways and the chemicals that they secrete in the brains of people with Down syndrome across the lifespan. And I think a lifestyle approach is going to be incredibly important here. Because there's going to maybe potential pathways that are in change over time, but are also up or down in younger individuals with Down syndrome. So Dr. Alessandra Martini, who was a project scientist in my lab, along with Alex Hellman, did a very careful study, extensive study, looking at different morphological phenotypes of microglia in the brains of people with Down syndrome and characterized them based on their shape, and looked at, for example, resting microglia, activated microglia. And what Alessandra noted was that dystrophic microglia, which had little blebs on them that indicates that they are probably dying or dysfunctional, that number, those types of microglial cells were the ones that we saw most frequently in the brains of people with Down syndrome. And it turned out right in parallel, and this is why I'm so happy Lisi has joined us in our lab, as part of her dissertation work in Montreal with Claudio Coelho, she essentially found the same observation, a whole different cohort of brains from people with Down syndrome. And then she did some beautiful measures of different cytokines and chemokines along with that. And all of that data come together to suggest we are seeing fewer resting microglia with age. Not surprising, that's what we see with aging and in Alzheimer's disease. But this increase in the number of dying microglia was really interesting to us, and could be a specific phenotype for people with Down syndrome. So where are we going from here? So there's lots of additional work that we need to do and chase down in terms of looking at vascular contributions to AD pathology, looking at their neuroinflammatory response, how microglia are responding. But also we're doing a lot of work in the astrocyte area in our lab. And certainly in a lot of studies with Down syndrome, there's still a lot of gaps in some of our basic knowledge that have been covered so elegantly in late onset Alzheimer's disease. So we're spending a little bit of time doing catch up. And, so there isn't a lot of work looking at S100 beta and the characteristics of astrocytes in the brains of people with Down syndrome. And now that there's all this beautiful work suggesting that there's homeostatic versus toxic astrocytes, we're very carefully trying to map through some of those markers to phenotype astrocytes in the brains of people with Down syndrome. I also have, and that work is being done by Leslie Vasquez-Rangel and Kevin Kamey. Now, Leslie's started graduate school at Duke University, so I'm really proud of her. And then Cherie Stringer is a graduate student in the lab who's developed a real interest and is working with Haley Miyasato, an undergraduate student, real interest in looking at aquaporin 4s. And given their role in potentially helping to clear beta amyloid from brain, this might be very, very important for people with Down syndrome. So Cherie is going to work through what aquaporin 4 receptors look like and whether or not they change their distribution on astrocytes in a pathological way, away from the blood-brain barrier where they're supposed to be. So she's working on some super high-res microscopy now with our brain donors, looking at those tissues. So I'm pretty excited to see what she's going to find in the future. So I can't talk about Down syndrome and Alzheimer's disease without doing a little pitch for our Alzheimer's Biomarker Consortium for Down syndrome. And I'm really excited about this project, and I wanted to bring it to people's attention because we are creating a huge data set from 550 people with Down syndrome that we're following every 16 months at various clinical and research sites across the country and one in Cambridge. And we're collecting clinical neuropsych data, tons of neuroimaging, amyloid, tau PET, FDG PET, and lots of MRI outcome measures such as ADAMS, and he'll talk about that tomorrow. But we're also collecting a lot of biofluids, much more limited cerebrospinal fluid, but lots and lots of plasma fluids. And as I hear some of these beautiful talks this morning, and I'm looking forward to the rest of the conference, all these pathways that are being described are potential targets as biomarkers. And so it's something I'd like to encourage folks to think about. And we're also collecting neuropathology data. And we have a nice collection of human brains from people with Down syndrome that include frozen and fixed samples. And I've heard a little talk about trying to confirm some of these elegant lines of research in iPSCs and in culture and in mice, and maybe taking a look in human brains. And so there's a number of groups that have tissue. And of course, I'm always very supportive of that. So I'll just end there. And I want to say very much thank you to our participants, and especially for families who consider participation in brain donation. This is incredibly important for us. And I think we always need to kind of go circular in terms of learning things from mice, iPSCs, to human brain, and then back again. And that's going to be the most powerful approach to finding new ways to improve quality of life for people with Down syndrome as they get older. I have an amazing team of people I work with here at UCI that helps collect information from people who are in our studies. But importantly, I wanted to acknowledge my lab. Of course, they're doing all the really beautiful work. And I also would like to acknowledge the sources of funding. The NIA, through the Include program, has been absolutely fantastic and helpful to us in developing ABCDS. And I also want to do a little shout out to the Bright Focus Foundation. They've helped support an international brain bank for people with Down syndrome that is trying to make more tissue available to researchers. And also supported some of our individual research projects. So thank you very much again for the invite, and I'm happy to take questions.
Thank you for that incredible talk, Professor Head. I'll open the floor to questions if anyone has them. Go ahead, Tracy.
Dr. Head, thank you so much. Can you hear me?
I can, yes, thank you.
Wonderful talk. And I had no idea that the numbers were so high that up to 15% of people who make it to age 70 actually with Down syndrome actually don't develop cognitive decline. And you said that wasn't due to partial trisomy, right?
Right. That was, that would be the first thing you might consider, is that the karyotyping.
So I guess I was just wondering about the rest of the genetics. And if that's something that you're looking into, if there's an effect of ApoE genotype, for example,
right.
All of the other known genetic risk factors for dementia.
Right. And yes, we are. So an interesting thing about the karyotyping is that we really have to lean heavily on karyotyping done in childhood, because there's increased mosaicism with age. And that can be a little misleading when we karyotype a little bit later. But we have a fabulous group at Columbia University led by Joe Lee. And Joe is doing GWAS on everybody and uploading that data, by the way. It's going to all, everything we do is publicly available upon, just a request form. And so Joe is kind of going through and looking at the common SNPs that we've identified in GWAS that might also affect risk and age of onset in our cohorts. And ApoE4, that's a really interesting literature. When you look at everything put together in studies of people with Down syndrome, that the effects can be really variable, that some report ApoE can, the presence of ApoE4 drives it earlier. There were hints of that in different papers. Some papers didn't see an ApoE genotype effect, but these were small kind of cohort studies. But I think one of the biggest studies that have come out now by Juan Fortia's group in Barcelona, Spain, does a really nice job and really shows convincingly that ApoE4 definitely has an impact here.
Makes sense.
Yeah.
No, it's super interesting. And I hadn't considered the mosaicism increasing over time also.
It does, yeah.
And so is there like a loss of the extra chromosome over time? Is that what you mean? Or is it more complex?
I honestly don't know, Trace. I'd have to see if Dr. Lee had some ideas on why that would happen. But, I think there's some evidence of that in other ways in aging and neurotypical people as well. So I don't think it's unique. Yeah.
Thank you so much.
There's a question in the chat. Elena Laughlin is asking, in the ABCDS database, are you collecting PBMCs and looking at immunophenotyping to see if there's a signature? From other studies, do you see increases in T cells near the vasculature that you're characterizing in these patient samples, similar to recent work from Tony Weiss-Correa? Right.
So as far as PBMCs and everything, a set of our samples go to the National Coordinating NCRAD, National Coordinating Center for Alzheimer's Research. And so those are samples, I believe, that can be requested through ABCDS. Biospecimens, of course, need a little higher level of review because it's an exhaustible resource. So we do have a proteomics core, an omics core on ABCDS. And there's some gorgeous work that Melissa Peterson is doing where she's finding that some of the best biomarker signatures for onset of Alzheimer's disease or MCI in people with Down syndrome is related to inflammation. Inflammation markers turn up every time as her top predictors. So, and she's published a nice paper on that. As far as the brain is concerned, I've poked at a little bit of T cell immunolabeling, but I think there's better ways we can look at this, and I think that's an excellent idea.
Thank you. Liwei?
Actually, Gil has a question. So Gil, why don't you ask first?
Yes, great talk. Thanks so much. It's interesting to think that senile plaques and CAA in a way compete with each other for amyloid deposition. And I guess the question is, there are some studies that showed that if you reduce the number of microglia with drugs such as CSF1R antagonists, the PTX drug, in amyloid models, you sort of reduce the amount of senile plaques and you favor the appearance of CAA.
Right.
And so with that notion, and also what you mentioned about perhaps a greater number of dying microglia in the DS, individuals with DS, do you think there's a possibility that it's a depletion of microglia that sort of may redirect that amyloid from the plaques to the CAA, just replicating in a way the preclinical studies?
I think that's brilliant. So we've been thinking about this a little bit with Matt Burton-Jones and his graduate student Sepide has done some beautiful work with fire mice showing exactly that effect, that she drives, when you knock out the micros, you drive away from plaques into CAA. And we're seeing these dying microglia. So it begs the question, is that the reason why we're seeing this? And, obviously knocking down microglia in people with Down syndrome might not be a good idea, which is another thing we might want to consider in future. Mm-hmm. Yeah, I love that observation. Thank you.
So do we have time for another question?
Yeah, go ahead, Liwei.
Okay, so the loss of parasites you show is really, really striking. That actually reminds me of Bessa Zokolov's publication in ApoE4 carriers. They also show lots of parasites, and that has a very good correlation with the BBB breakdown or leakiness. So do you see such a correlation in Down syndrome patients?
The short answer is I don't know yet. Lucy's working on that, and we do have brains from people who have E4. And I think some of the questions that are driving some of this project are based on his work, which is so beautiful. So, but you can see there's so many opportunities here. There's just so little out there. And so that's a great idea. We'll make sure to classify people into their categories or stratify them and see if we might not be powered enough, but we can at least say something a little descriptively about it.
Great, thank you.